I'd like to thank the BPS and ASCEPT for the opportunity to present today. I'm very honoured to be able to be here, and I'm also a bit humbled by it as well. The burden of heart failure should be familiar to most of this audience. In Australia, it's estimated that about 300,000 people have been diagnosed with heart failure. The problem is equally prevalent in the UK, and in fact, just before I got on the plane to leave Melbourne, there was an article featured on Twitter from the British Heart Foundation saying that about 920,000 people in the UK are thought to have heart failure. And this has actually risen by a third in just the last 5 years in terms of heart failure hospital admissions. And so this places a greater burden on health services than most other diseases do, because heart failure hospitalisations result in a hospital stay that's roughly double the average for all admissions. And indeed, the medical director of the British Heart Foundation was quoted as saying that heart failure poses a growing and increasingly complex challenge, not only for people living with the condition, but for those who care for them. And how we diagnose, treat and care for these patients could indeed be better. The increasing global prevalence of diabetes, and together with an ageing population, has actually created a heart failure epidemic. In that about a third of patients in clinical trials for heart failure have diabetes. Some of them might only be diagnosed at the time that clinical trial commences. And indeed, diabetes is an independent predictor of poor outcome in heart failure. And despite the higher rate of heart failure in people with diabetes, there's no specific treatment for heart failure for people with diabetes. So here are just some statistics around the problem of diabetes and heart failure. So the lifetime risk of diabetes now exceeds 1 in 3 of the population worldwide. And it's now estimated that by the year 2040, 642 million people around the globe will have diabetes. And how we can see that this is a problem in terms of heart failure is when we look at the cumulative incidence of hospitalisation or death due to heart failure. Here you've got the incidence in diabetic males and females, and you can see that even over a 3.5-year period, the males and females are pretty much on top of each other, whereas in the non-diabetic population, the incidence is about half that. So this just goes to show you the kind of burden that the combination of diabetes and heart failure represents. We've been coming at this from a few approaches, both pharmacological interventions and more recently from cardiac selective gene therapy approaches. And we've shown that an increase in reactive oxygen species in the myocardium is one of the causal contributors to diabetes-induced heart failure. And we've been able to show that constitutively active PI3 kinase alpha is a novel but interesting way in which we can counter-regulate diabetes-induced cardiac ROS generation. And we first showed this using transgenic mice that had a constitutively active PI3 kinase in the myocardium from birth. But that's a nice way of preventing heart failure, but what we were really interested in was intervening after the diabetes-induced remodelling and dysfunction was evident. So together with my colleague Julie McMullen, who's at the Baker Institute, we used a cardiac selective PI3K P110 alpha gene delivery in mice with type 1 diabetes. And what we were able to show firstly, in the diabetic mice that received an empty vector, there was a lower endogenous expression of PI3K alpha. We did not really overcome this with our PI3K gene therapy, but what you can see here is that this shows that it's cardiac selective. So here's the endogenous level of PI3K, and you can only see our AAV-delivered PI3 kinase, because it's a different size, in the left ventricular samples and not in lung, kidney, liver or skeletal muscle, showing that it really was a cardiac selective gene delivery approach. And we were able to show firstly that administering our PI3K alpha gene delivery 8 weeks after we'd induced type 1 diabetes, when we already see an impairment in cardiac function, could rescue the phenotype in the diabetic heart. So when the first endpoint that we looked at was markers of oxidative stress, superoxide generation and its sources. So when we looked at subunits of NADP oxidase, particularly NOx2 and P22-FOX, in the diabetic myocardium, here you can see the diabetes-induced increase in the null vector treated animals, but our PI3K gene delivery treated animals, shown here in black, the diabetes-induced increase in NOX2 and P22 FOX protein was completely prevented in the mice given the PI3K alpha gene therapy, with a similar benefit at the level of PKC beta 2. And this is one of the regulators of NADPH oxidase that is known to be increased in the diabetic myocardium. The level of diabetes-induced superoxide generation, estimated using lucigenin chemiluminescence, was also attenuated by our PI3 kinase AAV, as was NOX4 protein levels in the diabetic myocardium. This was able to also counter diabetes-induced cardiac fibrosis, whether we used collagen deposition, shown here with serious red staining. Here's the diabetes-induced increase in cardiac collagen levels. As you can see, there's much more red in these sections. This is completely prevented by the diabetic mice that received the PI3K alpha gene therapy, as you can see from the pooled data here. And we see similar evidence at the level of pro-fibrotic gene expression. Connective tissue growth factor is increased in the diabetic myocardium, and this is completely abolished in the mice given the PI3K gene therapy. We also see prevention of diabetes-induced apoptosis. Here's the diabetes-induced increase in the number of tunnel-positive cells in the type 1 diabetic myocardium, and this is significantly reduced in the mice given PI3 kinase alpha, and we see a similar trend at the level of the BAX to BCL2 ratio. The diabetes-induced increase is almost completely abolished in the mice given the PI3K alpha gene therapy. When we look at cardiac function, which is really the endpoint that we're particularly interested in, here's what we see in terms of diastolic dysfunction, which is the key hallmark of diabetes-induced impairments in cardiac function. This is reduced after 8 weeks of type 1 diabetes. Then we give our AAV gene therapy, and we follow these mice for a further 8 weeks. The mice that got the empty vector showed no improvement in the decline in EA ratio, whereas this decline was no longer significant in the mice that had been given the PI3K gene delivery. This was even more pronounced on 2 other markers of diastolic function, the diabetes-induced prolongation of deceleration time, which was blunted, and the diabetes-induced prolongation of isovolumic relaxation time, which was also blunted by PI3K gene delivery. We also saw benefit at the level of systolic function, the contractile function of the myocardium. This is something that happens later in diabetes-induced heart failure, as opposed to most other heart failure pathologies where the impairment in contractile function is what we see earlier. So after 16 weeks in total of type 1 diabetes, we see a modest but significant impairment in fractional shortening, which is certainly not evident in the PI3K diabetic mice. And we see similar trends at the level of velocity of circumferential fibre shortening, with a protective effect in the PI3K treated diabetic animals. So I've gone over this fairly quickly because this work was published a couple of years ago, but I'm sure what you're all thinking is, well, type 2 diabetes is really the problem that we're seeing in the clinic. This is where 90 to 95% of our diabetic population are. Surely that's more relevant. And the work I'm going to show you now has not been published. It's in second round revision at the moment at the American Journal of Physiology. And we've used the same PI3K gene delivery approach, but in a model of type 2 diabetes in mice. So these mice receive a high-fat diet for 26 weeks, combined with a low dose of streptozotocin to induce a mild type 2 diabetic phenotype. And after 18 weeks of this model, when we see an impairment in cardiac function, that's where we intervene, again with our same gene delivery approach of PI3K alpha. So what you can see here is that the mice are diabetic at endpoint, shown here with blood glucose and glycated haemoglobin. This diabetes phenotype at the systemic level is not affected by our PI3K gene delivery, nor would we expect it to. It's a cardiac selective intervention. We don't see any improvement at the level of glucose tolerance. This is the time for 2.5 hours after an acute glucose load, and here you can see the pooled data of the area under curve. But again, you can see that we have had cardiac selective transduction of our gene delivery. Here's our delivered PI3K alpha protein, and here's the endogenous level. And we're not seeing it in the skeletal muscle, the pancreas, maybe a hint in the liver. We're certainly not seeing it in the kidney or the lung. We then, again, we looked for markers of cardiac superoxide generation, firstly using lysogenin chemiluminescence, as I showed you previously. Here's the diabetes-induced increase in left ventricular superoxide, which is blunted in our PI3K gene-delivered mice. And here now we've confirmed it with a hydroaethidium fluorescence. As you can see here with the representative image, the red shows the superoxide generation in these left ventricles, and when we give the PI3K gene delivery, you see much less red intensity. And here's the pooled data over here, where we've been able to reduce the diabetes-induced increase in superoxide levels by about half with our PI3K gene delivery. We also see benefit at the level of both interstitial fibrosis and perivascular fibrosis. So we've looked at this under both polarised light and bright field, and you can see that the diabetes-induced increase in interstitial fibrosis and perivascular fibrosis, shown in this pinky red, is attenuated by our PI3K gene delivery, with the pooled data over here. So the diabetes-induced increase in type 1 collagen and type 3 collagen, which we can distinguish using the polarised light approach, and total collagen is significantly attenuated with our PI3K gene delivery, with a very similar result at the level of perivascular fibrosis. When we look at profibrotic gene expression, the diabetes-induced increase in CTGF and TGF-beta is completely attenuated by the PI3K gene delivery in our type 2 diabetic mice, and we no longer see a diabetes-induced increase in TIMP2 gene expression. We also look at hypertrophy of cardiac myocytes by tracing around the area of cardiac myocytes in H&E stained sections. And you can see that the diabetes-induced increase in cardiomyocyte width is completely attenuated by our PI3K gene therapy, as is the diabetes-induced increase in AMP gene expression. When we look at cardiac function in this particular model of diabetes, this is a model of mild type 2 diabetes, we didn't see any particular impairment in diastolic function, which surprised us, because we usually see that in this particular model, but we did see an impairment in systolic function, so in fractional shortening. So here we have 18 weeks of diabetes, where the diabetic animals are already seeing an impairment in fractional shortening. We give our gene therapy here and follow the mice for a further 8 weeks. The mice that were diabetic and received the empty vector, shown here in the black symbols, they continued to have a modest but significant impairment in fractional shortening. The diabetic mice that received the PI3K gene delivery, shown here in blue, showed a nice recovery in the fractional shortening measured on M-mode echocardiography, with a very similar result on velocity of circumferential fibre shortening, as you can see here, the improvement in the blue circles. So we thought that was pretty exciting. So this showed us that not only were transgenic mice protected against diabetic cardiomyopathy, which had PI3 kinase increase in constitutive activity from birth, but we could intervene after the diabetic cardiomyopathy was apparent with PI3K gene delivery, and we could also blunt both the diabetes-induced increase in ROS generation, but also the remodelling and the impairment in cardiac function. So then we turned our attention to something which we're referring to as maladaptive cardiac glucose metabolism, and a particular fate of glucose metabolism which upregulates ROS and is upregulated by ROS and plays an interesting role in the diabetic heart. And this is called the hexosamine biosynthesis pathway, or HBP. Only very small amounts of glucose go through the hexosamine biosynthesis pathway instead of glycolysis and other routes of glucose metabolism. It has been estimated it's somewhere between 2 and 5%, but this hasn't actually been accurately quantitated, but suffice to say it's a very small amount. But it goes through this pathway that results in a number of steps to create a metabolite called UDP-GLICNAC, which provides this GLICNAC as a substrate for post-translational modification of proteins. So GLICNAC will be added to proteins at serine and threonine residues, which, if you are interested in phosphorylation, may well be the very same site or an adjacent site and can actually interfere with phosphorylation of the same protein. The O-glicnacylated protein then has a change in the activity of that protein. Under normal physiological conditions, this is a protective pathway. which helps the cell to cope with any kind of acute cellular stress, such as a nutrient deprivation, hypoxia. It keeps cells alive, it preserves mitochondrial integrity, it preserves mitochondrial function and helps cells survive. However, diabetes is different for some reason, and we don't really understand why. But in the context of diabetes, this pathway is elevated pretty much throughout the body, and it stays elevated. And we think this is part of what is contributing to the adverse effects of diabetes on the heart. And hopefully the next few slides will show you that, will hopefully convince you. So this was shown or postulated in a review a couple of years ago, that we need to keep our levels of OGT and OGA, the two enzymes that control this GLICNAC modification. So OGT adds the GLICNAC to the protein and OGA removes it. This keeps the levels of GLICNAC in the cell at an optimal level. If you don't have enough nutrients, so in the cellular starvation state, you won't have enough GLICNAC, and that's not a good thing. That can lead to disease. But in the context of diabetes, where you have too much nutrient availability, you have too much GLICNAC modification, and that also leads to disease. So severe chronic insults to this O-GLICNAC buffering system can lead to loss of O-GLICNAC homeostasis and pathogenesis of disease, such as diabetes. So we've shown that this is something that's actually evident in the human myocardium. Firstly, we looked in atrial explants from patients who were undergoing bypass graft surgery, and we showed that the total amount of GLICNAC-modified protein in atria from diabetic patients, so this is not patients with heart failure, patients who had some sort of coronary disease, this was higher in the diabetic versus the matched non-diabetic patients, and this is what a Western blot looks like. There are supposed to be multiple bands because about 3,000 proteins are actually susceptible to this GLICNAC modification. But so this is a non-diabetic patient, and these are two diabetic patients. We also see that the gene expression of OGA, which removes the GLICNAC moiety, it tends to be lower in the diabetic myocardium. We then looked at left ventricular biopsies. After all, it's the left ventricle that does most of the work in the myocardium. And you can see, again, in the human left ventricle, the diabetic patients have higher amounts of GLICNAC-modified protein. And this actually is related to the levels of HbA1c, a marker of long-term glucose control. It's proportional to the elevation in blood glucose in our diabetic patients. And importantly, the more the amount of O-GLICNAC modification that's seen in the left ventricle, the lower the ejection fraction is. So cardiac function is worse in the patient who has a higher level of GLICNAC density. So we developed two opposing AAVs. One that expressed the human isoform of O-GLICNAC transferase. So this adds the GLICNAC moiety to the serine and threonine residues. And I'll show you these results, and then I'll show you the results for the opposing AAV, which was for OGA, to remove this GLICNAC moiety and return the protein to its native state. So again, we followed mice for 8 weeks of type 1 diabetes and then intervened with our AAV and followed the mice for a further 8 weeks. And this was the work, as was the previous two studies, of Darnell Precozo's PhD, and Miles co-supervised Darnell's PhD with me. So all of these next series of graphs show the white squares with an orange border as the non-diabetic mice given the AAV for OG2, the black squares the diabetic mice given the empty vector, and the solid orange squares are diabetic animals given OGT. And we actually expected that giving OGT on top of diabetes would actually have an additive effect. What we actually saw was giving the OGT to non-diabetic animals made the phenotype of those, the hearts of those mice actually look a bit like a diabetic mouse heart. So this is just showing that we got transduction of our gene delivery in both the non-diabetic and diabetic animals. When we look at this at the protein level, the mouse OGT has a slightly different size to the human isoform of OGT. So we see two bands on the mice that have got transduction of our OGT AAV. What we can see is that we were able to achieve a significant, modest but significant increase in the total amount of O-GLICNAC modified protein in the myocardium. It's only an increase of about 20%, but it's enough to have quite an impact on the myocardium, as I'll show you in subsequent slides. And we like to think of this as slightly turning up the volume of the amount of Glicknack modification in the myocardium, as opposed to a seven-fold increase. It's just a mild effect, but it's enough to induce an impairment in Glicknack homeostasis. This mild impairment increases cardiac superoxide generation by about 60%. Interestingly, it also increases expression of GFAT, which is the rate-limiting step of the hexosamine biosynthesis pathway, which is upstream of OGT, something we were not expecting. When we look at cardiac fibrosis measured under polarised light, you can see here this is the collagen that's lighting up on this image. When we look at type 1 collagen in the non-diabetic myocardium given the OGT AAV, we can see we've got a significant increase in type 1 collagen, not as much as we see in the diabetic myocardium given OGT, but certainly significant, and there's no additive effect in the diabetic mice given OGT. We also see an increase in type 3 collagen in the non-diabetic mice given OGT, and total collagen in the non-diabetic mice given OGT, again, is significantly increased. We see a very similar trend at the level of perivascular fibrosis, but now we see that the increase in type 1, type 3 and total collagen in the non-diabetic animals given OGT matches that of the diabetic animals. This mild change in Glicknack homeostasis is enough to have quite an impact on the myocardium. Firstly, if we look at diastolic function, you can see both groups of diabetic animals after 8 weeks of type 1 diabetes have a significant reduction in EA ratio. When we then intervene with the, so this is a non-diabetic mouse up here, this orange border white fill. This non-diabetic animal, given OGT at this time point, then shows a significant impairment in EA ratio. So we now see an induction of diastolic dysfunction. When we look at two other markers, deceleration time is significantly prolonged in the animals that are non-diabetic but given OGT, and we see a decrease in E prime A prime. So 3 different markers of diastolic function are impaired in the non-diabetic animals given a cardiac selective OGT AAV. We also see a modest but significant impairment in systolic function, so fractional shortening. When we do the converse experiment and we give the human isoform of OGA, after 8 weeks of type 1 diabetes, we are actually able to rescue the cardiac phenotype in the diabetic mice without any impact on the non-diabetic mice. So in these studies, we've given 2 different doses of the AAV. The same dose we gave of the OGT, and then a dose that was 5-fold higher. So you can see that if we give a 5-fold higher dose, we get about a more than a 5-fold increase in the amount of OGA transduction, and certainly more than 5-fold higher the amount of protein. This is the OGA that we've given by gene delivery in the non-diabetic and the diabetic myocardium at the normal dose, and there is a band there. It's just that the band for the ones given 5 times higher dose is much stronger. So we're able to show that a 5-fold increase in the dose of gene delivery has a much more remarkable effect at the level of transduction. What we see is here's the diabetes-induced increase in LV Glicknack modified protein. This is attenuated by both the lower and the higher dose of OGA gene delivery. This is also able to counter the diabetes-induced increase in superoxide generation in the myocardium. So now we've been able to turn the volume back down a bit in the diabetic myocardium. And this is able to rescue the cardiac phenotype. So here's the diabetes-induced increase in type 1 collagen, interstitial type 1 collagen. This is attenuated by both doses of OGA. Similarly for type 3, and the total amount of interstitial fibrosis induced by diabetes is almost completely abolished by both doses of our OGA. We see very similar trends at the level of perivascular fibrosis. Here's a diabetes-induced increase in type 1, type 3 and total collagen being markedly attenuated by both doses of OGA. We also see similar benefits at the level of hypertrophy of cardiac myocytes. Here's the diabetes-induced increase in cardiomyocyte area and cardiomyocyte width, and both are attenuated by both our our lower and higher doses of OGA. What we did note, however, was that we didn't see a lot at the level of gene expression for both fibrosis, so C2GF and TIMP2, or hypertrophy, AMP and beta-myosin heavy chain. All of these were increased in the diabetic myocardium, but seemed to be impervious to our OGA gene delivery. However, what we saw at the level of protein and the level of function was certainly of benefit. So here we see, before the AAV, the black symbol is the diabetes-induced impairment in EA ratio. In the mice given OGA, after we measured EA ratio at this time point and followed for a further 8 weeks, we see quite a profound rescue of the diabetes-induced impairment in EA ratio. We see rescue of the diabetes-induced prolongation of deceleration time, and we see rescue of the diabetes-induced impairment in E'(A'). We also see a tendency for an enhanced fractional shortening at both doses of our OGA. So we're able to see an improvement in left ventricular function just by restoring GLICNAC homeostasis. So then we wanted to look at what was the mechanism of the detrimental effect of the OGT and the protective effect of OGA. And you'll recall from the first 2 studies that I told you about that PI3K alpha is protective in the diabetic myocardium. So that was the first thing we looked at. So the non-diabetic mice, given the OGT, showed a significant impairment in PI3K protein levels in the myocardium and a significant impairment in AKT phosphorylation, which is the downstream mediator of PI3 kinase alpha. We also saw changes at the level of GLICNAC-modified proteins, and we chose 3 in particular that we were interested in. So firstly, we were able to show that OGT significantly increases GLICNAC modification of PI3K alpha in the heart. It increases GLICNAC modification of AKT in the heart, and excitingly, it also increases GLICNAC modification of the calcium handling protein SERCA2A, which is important for contraction and relaxation in the myocardium. So this might be one of the mechanisms by which OGT is detrimental in the diabetic heart. We show the converse scenario for our OGA. The diabetes-induced impairment in PI3K alpha protein in the diabetic heart wasn't affected by our OGA AAV at either dose, but the higher dose was able to enhance AKT phosphorylation in the diabetic myocardium. The diabetes-induced increase in GLICNAC-modified PI3K alpha, GLICNAC-modified AKT, and GLICNAC-modified SERCA2A were all completely attenuated by both doses of our OGA, the lower and the higher dose. So that might be one of the mechanisms by which our OGA AAV is protective in the diabetic myocardium. This is just to show that neither OGA or the OGT AAV had any impact on blood glucose levels or on body weight because it was specific to the myocardium. Then we wanted to see whether the mechanism was related to any impact on mitochondrial function in our diabetic animals. And so we first looked at this using the OxPhos kits, where you can measure all 3 complexes of the electron transport chain in the one Western blot. And we didn't really see a lot to get excited about here. Diabetes tended to reduce complex 3 activity, wasn't really improved by either dose of our OGA. Diabetes also tended to reduce complex 5 activity, which again, wasn't particularly affected by either dose of our OGA. So we decided to look at this a different way. We were able to obtain human iPSC-derived cardiac myocytes, and we were able to measure mitochondrial function using the Seahorse bioanalyzer. And we looked at both glycolytic activity using ECAR, and we were able to look at oxygen consumption, so mitochondrial respiration, using the Seahorse. And we did this in 2 different iPSC lines to confirm that what we were seeing was real. So firstly, at the level of glycolytic activity, so the low glucose incubated iPSC-derived myocytes are shown in blue. The high glucose treated iPSC-derived myocytes are shown in red, and you see an impairment in glycolytic capacity and an impairment in glycolytic reserve in our iPSC-derived myocytes that are incubated under high glucose conditions. However, the OGA AAV, shown here in the hashed bars, had no effect on this glycolytic capacity. However, when we looked at mitochondrial respiration, you can see that the high glucose-induced impairment in maximal respiration was almost completely prevented by our OGA AAV in the two different lines that we studied. So perhaps preservation of mitochondrial function was also another mechanism by which our OGA was protective. So what I've been able to show you is that in the absence of diabetes, if you increase the amount of OGT in the myocardium, you get Oglic-Knack modification of PI3K alpha and of AKT. You also get an increase in cardiac ROS levels, cardiac fibrosis, cardiomyocyte hypertrophy, and impairment in cardiac function. In the context of diabetes, the OGT doesn't make anything worse than what diabetes has already done. However, OGA is able to prevent the diabetes increase in Oglic-Knack modification of PI3K alpha, in Oglic-Knack modification of AKT, and I haven't shown it here, but in the diabetes Oglic-Knack modification of SERCA2A. And as a result, we're able to counter the effect of diabetes on cardiac ROS, mitochondrial function, fibrosis, hypertrophy, and cardiac function. So we then now speculate that the OGA, the Oglic-Knack A's, also counter-regulates cardiac ROS generation in the myocardium, and that's one means by which it's protective, as well as at the level of mitochondria and at the level of individual proteins that are Oglic-Knack susceptible. But just to show that not everything we do is around gene delivery, we also use some pharmacological tools. Where we've looked at mitochondrial-targeted methyl glyoxyl sequestering compound, mitoGA-mide, and this is one of the mitochondrial-targeted compounds developed by Mike Murphy at Cambridge. And this was a short study we did in type 1 diabetic mice, and we were able to show that Akita mice at 6 weeks of age, if we gave them the mitoGA-mide or vehicle for 10 weeks, we were able to see improvements at the level of diabetes-induced cardiac function. So what we were able to show was in the Akita type 1 diabetic mice, the reduction in peak A wave velocity on echocardiography, in peak E prime velocity on tissue Doppler was prevented by the mitoGA-mide. We didn't see any impact on A prime wave velocity, but the diabetes-induced impairment in E prime A prime was completely protected in the mice given the mitoGA-mide on tissue Doppler echocardiography. Very similar effects at the level of Doppler flow echocardiography. The diabetes-induced decrease in E wave velocity was no longer evident in the diabetic mice given the mitoGA-mide, and the diabetes-induced impairment in EA ratio was also no longer evident in the diabetic mice given the mitoGA-mide. We did see prolongation of deceleration time. That was tended to be reduced in the mice given mitoGA-mide, and the E over E prime ratio in the diabetic animals was also significantly reduced in the mice given the mitoGA-mide, suggesting that this might actually be a pharmacological approach to protecting diastolic function in the diabetic myocardium. We then have some unpublished data to suggest that we might be seeing benefits at the level of mitochondria. We've looked at the number of lipid droplets in the diabetic animals. This is reduced in those given mitoGA-mide. The N is very low here, which it's 2. We can talk about this later as to why the N was so low. We also saw changes at the level of gene expression for PARC2 and USP30. These are genes that are important for controlling mitochondrial autophagy. And we also saw an impairment at the level of P22-FOX gene expression in the mitochondrial samples. So what I've now shown you is that not just OGA is protective against diabetes-induced cardiac dysfunction, but so is mitoGA-mide. And we're now undertaking studies in a mouse model of type 2 diabetes, as we are with our human OGA studies, to see if we can protect the type 2 myocardium over the longer term from this diabetes-induced impairment in cardiac function. So this journey is still underway. We're also targeting diabetic cardiomyopathy by looking at HDAC4 inhibition, at looking at a range of antifibrotic interventions as well, but hopefully we're getting somewhere towards improving cardiac function in the context of diabetes. So on that note, I'd like to thank all the people that have been in my lab, who are all coming across to Monash with me at the start of the new year. We have a lab mascot, Barry, and I'd like to thank our funding bodies, and the most important thing, I'd like to invite you all to Perth this time next year to come to our ASCEPT APSA annual general meeting. You can have a lovely time surfing, going to the wineries, and of course some fantastic pharmacology. Thank you.
Thank you, Rebecca, for that fascinating presentation. I think we have a couple of minutes for a couple of questions, if anyone had any questions.
Hi, Rebecca. Very nice. Adrian here from Signature Discovery. That was really beneficial for heart failure in the context of diabetes. Is this generally applicable to heart failure?
That's such a fantastic question. So before we started thinking about this in the diabetic context, it was always thought that gliclanac was cardioprotective, and mice that have complete knockdown of OGT actually do worse after MI rather than better, but that is mice that don't have any OGT at all, as opposed to just turning the volume back down. Conversely, if you overexpress OGA in the myocardium in any setting other than diabetes, they haven't seen the protective benefit that we've seen, but they haven't followed the mice for as long as we have. They've looked at perhaps a week after myocardial infarction. They haven't followed them for, say, the 8 to 12 weeks that you'd need to see changes at the level of a real heart failure in terms of impairment in contractile function. We think there's something unique about diabetes because the high glucose levels are making glycanacylation go up, as is superoxide, and the high glucose is driving up the superoxide. We think there's some sort of vicious cycle happening, a nasty feed-forward loop that's making everything so much worse. That's what we're working on at the moment. We wouldn't want to be completely removing gliclanac modification from all proteins throughout the body. I think that would be detrimental. We want to just be able to control it fairly exquisitely.
I believe there's another question over there.
Okay, I've got three rapid-fire questions. One is, what does lipid metabolism change? That is, does acyl carnitines, etc., change in parallel with the glucose metabolism?
Probably. We also know that lipids can actually drive gliclanac modification. And so that's one of the things we've got next on our list to do, as is looking at what's happening in terms, right at the level of mitochondria, in terms of gliclanac modification of specific proteins inside the mitochondria. We want to see what's changing there as well. We feel like we're just at the tip of the iceberg here, and we've got so much more to uncover.
And streptozotocin doesn't affect the enzymes?
Yes. So if you directly add streptozotocin to isolated cells, I think it impairs. It does affect the pathway acutely, and I'm trying to remember whether it inhibits OGA or OGT. It inhibits one, and aloxan, which is also pro-diabetic, inhibits the other. So there seems to be something that's not related to diabetes-induced cardiac dysfunction, but a specific effect of both streptozotocin and aloxan having opposing effects on this pathway, and I can't remember which inhibits OGA and which inhibits OGT. But we definitely see upregulation of gliclanac modification in DB mice. So when we finish our current study in our high-fat SDZ type 2 diabetic mice with our AAVs, our next step is to take this into DB mice to specifically look at a model of type 2 diabetes that is not only more severe but is independent of streptozotocin to specifically get at that.
So one quick
one, and we'll wrap it up there.
Which is that it's a big target in Alzheimer's, which appears to go the same way. So, therefore, how beneficial will it be just to have cardiac aspects in diabetes?
Well, I think you could also approach a brain selective approach as well. We just wanted to see if we were specifically getting benefit that was independent of any hemodynamic effects outside of the heart, to really understand what was happening at the level of the myocardium. We're a long way short of being able to develop a drug to manipulate oglycanic modification of specific proteins, and I think that's really what we'd want to be doing. It may well be something that's relevant in Alzheimer's, and there are papers in the literature that's actually referring to Alzheimer's as type 3 diabetes. So maybe there is a role for glycanic modification in Alzheimer's as well, but we haven't been looking at that.
Is there another question back there? Yeah.
Hi, Rebecca. I'm Rita over here.
I can't see
Right,
where you are.
back.
I'm Rita.
I'll stand up.
Oh, there you are.
Hi.
The lights are
I very much enjoyed your talk. I don't know whether I missed it. So, do any of these strategies work in reversal? When's the treatment time? Did I miss it? Is this pre-treatment or...?
No, it's definitely reversal. So for our type 1 diabetic animals, we wait till they're 8 weeks diabetic and we do the echo to see whether we've got an impairment in cardiac function. Then we give the AAV. So we are definitely looking at reversal in terms of cardiac function. I can't tell you whether any of the other changes, say like in fibrosis, are reversal or not, because we haven't assessed fibrosis partway through. We know that mice do have fibrosis after 8 weeks of type 1 diabetes, but we haven't looked in these animals. In the type 2 diabetic model, the animals have diabetes for 18 weeks because it takes a lot longer to see an impairment in cardiac function in our model of type 2 diabetes. That's when we give the AAV. So we are really trying to see reversal in the cardiac phenotype rather than prevention.
Thank you.
Okay, do we have one last question over there? Thank you.
Thank you. It's kind of similar to what has been asked. As we all know, prevention is better than cure. And I was wondering, have you considered taking that direction? Because it looks like you've done so much work to find a cure that has not been created yet. And I'm wondering whether you've looked into preventing the actual disease.
Oh, yes.
So if you intervene fairly early in streptozotocin-induced diabetes with insulin to restore insulin to its normal levels, you will prevent the entire diabetic phenotype. The problem is that in humans, their blood glucose levels are generally not as well controlled as they would be in a mouse, and not all glucose-lowering therapies are actually protective against cardiovascular complications. The newer glucose-lowering approaches appear to be effective, at least the SGLT2 inhibitors are showing some promise for heart failure, but almost any other glucose-lowering intervention has not shown any benefit on heart failure. Even the GLP-1 receptor agonists, they're effective against atherothrombotic events, but we're not yet to be convinced about whether they're protective against heart failure. So the patients are also the impairment in cardiac function that's seen in diabetic patients can actually be seen in about 70% of patients with well-controlled diabetes who don't have any impairment in blood pressure, who don't have any obvious cardiovascular abnormalities, will already have an impairment in cardiac function. It's just you have to go looking for it. It's clinically silent.
Okay, thank you, Rebecca. I think it's been a fascinating session today, and please join me in thanking Rebecca and her fascinating talk today.